welcome to regulatory requirement for medical devices including in vitro diagnostics in India version 2. Lecture 5 that is regulatory requirement of the biocompatibility of medical devices as per ISO 10993. Learning objective of this lecture: be aware what is the biological evaluation test, what is the requirement of this biological test on the medical devices, expected outcome. The participants can able to understand what is the requirement of the biological evaluation of the medical devices, what standards is applicable for the biological evaluation, and the regulatory provisions for the requirement of biological evaluation test for medical devices. Target audience: the personnel working in the medical devices and in vitro diagnostics industry, the startup involved in the medical devices and diagnostic industries, the regulatory affairs personnels, human ethics committee members, clinical trial team members, researchers, students, academicians, and the person generally interested in the field of medical devices and in vitro diagnostics. What will we learn in this lecture? We will cover: first we will discuss what is the biocompatibility, then bioevaluation with risk management process, what are the process for risk management process for the biological evaluation, then categorization of the medical devices and test selection based on the category of the medical devices, which type of biological evaluation is required, the standards applicable for conducting the biological evaluation, and the general principles of the standards prescribed for biological evaluation of the medical devices. So, all this topic we will discuss in these lectures, and we will have, after detailed discussions, we will have the question answer session. Now, what is the biomaterials? Biomaterials can be derived either from the nature or synthesized in the laboratory using the variety of the chemical process, variety of the approach utilizing the metallic component, polymers, ceramics, or composite materials. They are often used and adopted for medical applications and perform and replace the natural functions of the human body or augment the natural functions. This biomaterial may also be an autograft, allograft, xenograft used as a transplant materials. The purpose of performing biocompatibility testing of these biomaterials is to determine whether the suitability of the devices for human use and to see whether use of the devices can have any potential harmful physiological effect on the human body. So, this is the purpose of the biocompatibility. The material characterization and analysis of the devices components are conducted prior to conduct of any biological evaluation. It is not key once the biomaterial is developed, immediately the biological evaluation of biocompatibility test can be performed. Prior to conduct of the biological evaluation, the material characterizations and analysis of the devices is compulsory. This material characterization includes extracting leachable materials from the devices or the component at the elevated temperature and analyzing leachable extract for potential harmful chemicals or cytotoxicity. Once this in vitro testing has completed, the in vivo biological test can be done based on the device intended use. The biological evolution testing can be ranges from the skin irritation test to hemocompatibility test, implantation test, etc. The turnaround time for biocompatibility test can ranges from 3 days to greater than the several months, depending on the specific test data needed for the materials. The subchronic or chronic implantation test can last even longer period. The two ways to test the biocompatibility in vivo implants are tissue culture test and blood and contact test. The example of the biomaterial applications in the medical devices, we have discussed what is the biomaterials and the applications of the biomaterials. These biomaterials applications is meant for manufacturing of that joint replacement, bone plates, bone cements, artificial ligaments or tendons, heart valves, skin repair devices, breast implants, vascular grafts, stents, nerve conduits, contact lens. These are the applications of the biomaterials, and which biomaterials are generally used for which applications, we can discuss that. The various biomaterials are available and established. The like silicone rubbers, this material is to be generally used in the manufacturing of the catheters or tubes. Cellulose, that is for the dialysis devices. Polyurethane, that is used in the manufacturing of catheters, pacemaker leads. Stainless steels, metals, if you talk about the metals, the stainless steel that is used in the orthopedic devices or cardiac stent. Titanium, that is also used in the orthopedic implants and also in the dental devices. Alumina, orthopedic and dental devices. Hydroxyapatite, that is also used in the orthopedic or dental implants. Collagens, ophthalmological applications or various wound dressings. The collagens are to be used. So, these are the applications of the certain biomaterials which is used for applications of the manufacturing of this type of the medical devices. Now, come to the definition, certain definitions that related to the biocompatibility. First of all, what is the biocompatibility? Biocompatibility is the measurement of how compatible a device is with the biological system. Another way we can say it is the ability of the device material to perform with an appropriate host response in a specific situation. Now, the other definition, that is direct contact or indirect contact. What is the indirect contact? Indirect contact is defined as the term that used for the devices or device component that comes into physical contact with the body tissue. If it is physical contact with the body tissue, it is known as it is considered as a direct contact. What is indirect contact? The device or device component through which a fluid or gas passes prior to the fluid or gas coming into physical contact with the body or body tissue. So, it such type of that devices or the device component through which a fluid or the gas passes prior to fluid or gas coming into the physical contact with the body tissue, that can be considered as a indirect contact. Now, biological evaluation, it is also known as the biocompatibility. The biological evolution of the medical devices is performed to determine the potential toxicity resulting from the contact of the component material of the device with the body. This is the biological evaluation. Now, finished form of the devices, that is the final finished form. This term is used for the devices or device component that includes all manufacturing process including packaging, sterilizations if applicable. Novel materials, this term we can define as the materials that has not previously been used in any medical devices. New materials which is yet to be used in the particular applications. So, any new materials that has not previously used in any medical devices is known as, is termed as novel materials. Sponsor means manufacturer, application submitters who will submit the application for regulatory approval or any purpose to the authorities, that is termed as sponsor. Now, come to the biocompatibility. When biocompatibility is considered, we have to understand in which cases this biocompatibility test or the biological evolution test can be considered. It is critical for the regulators determination of the safety and effectiveness of the new medical devices or the modified medical devices. So, for any new devices, if the medical devices material come direct or indirect contact with the human body, then the biological evolution can be considered, or if the particular changes have been made in the devices which affect another component which has come to the tissue contact. In such cases, this biological evolution or biocompatibility test is also required. Modified devices, why it is required? Because in certain cases, if the new internal component is added on the particular medical devices, that added through some heat is to be applied to join the another component with the main devices. The heat could change the chemistry of the biomaterials. So, in such cases biocompatibility test is required. This biological evaluation or the biocompatibility test is also required for submission of regulatory approval for import or the marketing permissions or manufacture and marketing permissions. Sometimes this study is required to determine the potential for unacceptable adverse biological response. So, in such cases this biological evolution test can be performed on the medical devices or the component of the medical devices. Now, the biocompatibility test, the test should be carried out as per applicable standards. Some biological evolution test standards have been prescribed in pharmacopoeia, some have been described, certain guidelines or the standards have been described in other standards, but the ISO 10993 that is the uh, uh, standards developed by ISO for evaluation, for biological evaluation of the medical devices. Various series of these standards are available, and each series have different standards, guidance, principles that is included in the various series of the ISO 10993. Also some ASTM or ICH or OECD guidelines or standards are also applicable, and that is adopted by the other regulators. We will discuss that the standards ISO 10993, which is applicable for determination of the biological evolution of the medical devices as per our medical device rule 2017. So, in India these standards is in the law, it is clearly defined that ISO 10993, where the biological evolution has been prescribed, that should be applicable for conduct of this test. So, what are the main component, principles, guidance and approach for conduct of the biological evolution? We will have some brief discussion on these standards. What is the standards? This ISO 10993, these standards include consideration of the design devices, material component and manufacturing process. This also includes the clinical use of the medical devices, including the intended anatomical locations where it is to be implanted or used, the frequency and duration of the exposure, if the device, if certain time it has to be exposed to the human body. So, based on the frequency and duration of the devices, the test can be decided. Potential risk from the biocompatibility perspective, if there any potential risk is there, in such cases also these standards, this includes the consideration of such cases also. If certain information is available to address the identify the risk, any other risk, and the information needed to address any remaining knowledge gaps, if certain gaps are there in particular materials such as the new biocompatibility testing or the other evaluation that appropriately address the risk. In such cases, what type of that evaluation is required to be carried out on the biomaterials or the component of the medical devices or the medical devices, all those parameters that standards includes. The biocompatibility test may not be needed if the device is made of the material that has been well characterized chemically and the physically, and also there is a lot of published literature is available and have long history of safe use. In such cases, the bioevolution test cannot be required or cannot be considered, provided the adequate literature and the data on the product or the biomaterial has to be produced by the manufacturers to justify whether this is required or not. If the material or the manufacturing information is provided to demonstrate that no biocompatibility concern is exist on the medical devices. So, in such cases it is not necessary to carry out the biological evaluation test. If you see the risk based approach, the biological evaluation may be possible to leverage the previously conducted biocompatibility informations if the previously tested device have similar indication, similar type, and also the duration of the contact of the devices is the same. In such case also there is the biocompatibility study is not necessary. The information provided by the manufacturer to explain why difference are not expected to impact the biocompatibility. It means the manufacturer has to justify properly, adequately by way of the published literatures, the study already performed earlier, and the safe use, history of the safe use of that devices. They can justify for not performing the biological evaluation with the particular medical devices or component of the medical devices. Now, come to the biological evaluation. We have already discussed what is the biological evaluation. It is a part of a structured biological evaluation program within the risk management process in accordance with the ISO 14971. Risk management process, that ISO 14971 is applicable for all the medical devices and in vitro diagnostics. So, this biological evaluation study is the part of the risk management process in accordance with the 14971 ISO. This ISO 10993 series, we have discussed that it has various series and approximate 20 series of the standards is applicable. 20 to 22 standards are presently applicable for the biological evaluation. It is a high level guidance and how to conduct the biological evaluation, various types of the biological evaluation, that procedure, methods or guidance has been given in these standards. If you see the series 1, that is the general biological evaluation standards, it is the general standards. Here the details of the test method for the investigation of the different aspect of the biological safety, that is given. The annexures, it has 3 annexures, annexure A, annexure B, annexure C. Annexure A, the matrix is available, is given based on the categorization of the medical devices, which tests are required to conduct and which are not required, that annexure A is there. Annexure B, in which cases that how to approach the risk based analysis of the devices, and based on that what the study is required, that manufacturer can decide. Annexure C, that is the literatures, published literatures and the data review, how to review that available data with respect to the biocompatibility of the particular biomaterials or the medical devices. So, these annexures provided in this is ISO 10993 series. We will discuss that. We have discussed that ki 10993 series 1, that is the general standards which describes the general principles of the biological evaluation, the categorization of the medical devices based on the nature and the duration of their contact with the body. This categorization is given in this ISO 10993 part 1. The evaluation of the existing relevant data from all the sources. This describes the identification of the gap, that based on the standards, that based on the available informations, the identification of the gaps in the available data on the basis of risk analysis that can be carried out. The identification of the additional data set necessary to analyze the biological safety of the medical devices and the assessment of the biological safety of the medical devices. This component has been described in this first series of the standards. Annexure A already we have discussed, sorry. Annex A contains the informative tables. It is applicable that in general helpful for identifying the biological data sets recommended for evaluation of the medical devices according to the category of the body and the duration of the exposure of the devices to the human body. The appendix B, that is the guidance for the application of the risk management process, how the risk management process is applied. The biological evolution shall be planned, carried out and documented by the knowledgeable and experienced professionals. Annexure C, that is the suggested procedure for the literature review, how to perform the literature review for the existing data. These are given in this annexure C of these standards. This ISO 10993 does not cover the testing of the materials or the devices which do not come into contact or in direct contact with the patient body, nor does it cover the biological hazards arising from any mechanical failure. It means that these standards, this biological evolution test is only applicable to those devices which are direct or indirect contact with the patient body. Now, the risk approach to the biological evolution of the medical devices as a risk management process, we can discuss this. This flow chart you just see, in this flow chart it is clearly that risk management process has been applied, which cases this biological evolution is required, in which cases it is not required, and where all the biological evolution is required to be carried out. This flow chart gives that detailed idea. If you see the devices, the manufacturer can decide if the devices have direct or indirect contact with the human body or not. If it is in direct contact or indirect contact with the human body, then this standards is applicable and the biological evolution can be carried out on such type of the devices. If it is not in direct contact or indirect contact with the human body, the scope of this standard is not applied. That is, in such type of devices, this biological evolution test is not required to be carried out. If that biological evolution, that biomaterials is in direct contact with the human body, direct or indirect contact with the human body, then the device identifications, chemical and physical categorization of the devices shall be carried out by the manufacturers, and also after carrying out the chemical categorization, the requirement of the biological evolution that can be decided. We have discussed in the previous slides, prior to conduct of the biological evolution, this chemical categorization of the medical devices or the biomaterials is required to be carried out to prove the safety of the devices. So, once it is conducted, then the manufacturer has to see whether the materials, the biomaterials is the same which is commercially available in the market. If it is commercially available, then whether the devices have the same chemical component. If it is a same chemical component, then also he has to see whether the manufacturing or the sterilization, the manufacturing and sterilization of the devices, the method applied for the manufacturing and the sterilization is the same which is already available medical devices. If it is same, then whether the body contact is also the same. If the commercially available device have the same sterilization process, same chemical composition, same body contact, in such cases the manufacturer has to perform the biological evaluation assessment as prescribed in Annexure B of the standards. They will carry out the risk management process based on the data available, and after that they have to decide whether which test is required and whether the test carried out by the firm is sufficient to decide whether the product is safe for use in the particular applications or the device is safe for use in the particular intended purpose. If the manufacturing process, commercially available devices, sterilization process or body contact is not the same as available in the market, in such cases again they have to determine whether the sufficient data is available, sufficient justification is available, relevant data is there, the chemical and the biological data of the medical devices or the biomaterial used in the medical devices is there. If it is there, that is sufficient for the risk assessment. If it is sufficient for the risk assessment, then they have to see whether sufficient toxicology data exist for all the chemicals or the materials used in the biomaterials or the medical devices. If it is there, then the data, whatever the data that is applied to the chemical mixtures used in the biomaterials, if the relevant data are there, so the data is also same to justify that it is based on this data the assessment can be determined, and if all such data is available, then they have to perform the biological risk assessment as per the Annexure B of these standards, and particular test based on the biological risk assessment the manufacturer can perform and submit to the authority for the regulatory approvals or to establish the safety of the material or the medical devices being manufactured by the firm. If there is no sufficient justifications or the relevant data of the chemical or the biological data of the medical devices is not available, then the manufacturer has to carry out the biological evaluation. They have to select the biological evaluation test as per the Annexure A of these standards. Annexure A, which gives the details of the biological evaluation test carried out based on the categorization of the medical devices. So, that matrix can be adopted by the manufacturers, and based on that matrix the test prescribed in the annexures can be performed. The ISO 10993 Annexure 1 Annexure A, that is the minimum test required to carry out the devices or the biomaterial for which there is no sufficient justifications or the relevant data with respect to the chemical or the biological information is available, but other tests also can be carried out based on the nature of the devices. So, that is the minimum test that has to be performed by the manufacturer in case there is no adequate justification, relevant data regarding the chemical or the biological is available with respect to the medical devices or biomaterials. And after carrying out the biological evolution as per the annexure A of the standards, they have to justify ki in based on the data generated on the medical devices, the product can be safe for use in the particular applications or use for the particular intended use of the devices, and they have to submit the data to the authority for regulatory approvals or regulatory compliance. These are the principle or approach for conducting the biological evaluation, and it is clearly mentioned in the standards. This is a standard that is 109931, and this standards also recommended that the material categorization are very important steps prior to conduct of the biological evaluation of the medical devices or the biomaterials, and the material categorization is carried out, that is based on the standards of the ISO 10993, that is series 18 and series 19 of the ISO standard 10993 describes the details of the material characterization process methods. It is clearly mentioned in the this series 18 and 19 of the standards. So, based on that standards, the material categorization is to be carried out prior to conduct of the biological evaluation, and based on this material categorization, the data they can use for the further biological evaluation test for the medical devices. The extent of the chemical categorization or the material categorization depends on what preclinical or the clinical safety and toxicological data exist on the devices or the materials, the nature and duration of the body contact with the medical devices or the materials, but this categorization shall address the constituent chemicals of the devices and possible residual process aids or additives used in the manufacturing, and the details of the material categorization is prescribed in the series 18 or 18 and 19 of the ISO 10993. The various series of the biological evaluation is available so far, the published. There are approximate 22 standards series are there, ISO 10993. The series 1 that we have discussed in detail, that is for the evaluation and testing within the risk management process. This is the general series. Series 2 of this standard, that is animal welfare requirement. Series 3, that is the test for the genotoxicity, carcinogenicity and reproductive toxicity. Series 4, that describes the selection of the test of the medical devices that interact with the blood. Series 5 describes the test for the in vitro toxicity, how to conduct the in vitro cytotoxicity test. The methods have been described in these standards. Series 6, that is the test for local effect after implantation of the devices. Series 7, that is that the standard relates to the ethylene oxide sterilization residuals. Series 18, it is the selection and qualification of the reference materials for the biological test. However, this series has been withdrawn by the agency, it is no more is applicable. Series 9, that describes the framework for identification and quantification of the potential degradation products. Series 10, that describes the test for irritation or the skin sensitization test. Series 11 of this standard, that describes the test for the systematic toxicity study. Series 12 describes the sample preparation and the reference materials. Series 13, that is the identification and quantification of the degradation product from the polymeric medical devices. Series 14, that is identification quantification of degradation product from the ceramics. Series 15, that is the identification and quantification of the degradation product from the metal and alloy. Series 16, that describes the toxicokinetic study design for degradation product and leachables. Series 17, that describes the establishment of allowable limits for the leachable substance. Series 18, chemical characterization of the materials. We have discussed that. This series 18 and 19, that describes the chemical categorization of the materials, physiochemical, morphological or topographical categorization of the materials. Series 20, that is the principles and method for immunotoxicology testing of the medical devices. These are the other series of the standards which describes different that component, different test that is required to be carried out as a part of the biological evolution of the medical devices. Now, we will discuss the categorization of the medical devices and the test selection. We have discussed earlier that categorization of the medical devices as per the standard ISO 10993, that is based on the contact of the devices or the duration of the devices with the human body. And based on this categorization, which test is required to be carried out for biological evolution of the biomaterial or biocompatibility, that can be determined. So, categorization of the medical devices, already we have discussed that can be the medical devices can be categorized to facilitate the selection of the appropriate biological test based on the nature of the body contact and duration of the body contact. If you see the nature of the body contact, that device can contact with the surface of the body. Certain devices that contact that external communicating devices, and certain devices that is the implantable devices. So, three different nature of the contact of the devices, that is the surface contact, external communicating and the implantable devices, and duration of the contact, that is defined as a limited duration of the contact, prolonged duration of the contact or permanent duration of the contact. So, limited contact, prolonged contact and permanent contact duration, that is defined in terms of the duration, that if it is in body contact up to 24 hours, it can be considered as a limited contact. 24 hours to 30 days, it can be categorized as prolonged contact, and the permanent beyond 30 days if the particular device is implanted or in contact with the human body, that can be considered as a permanent contact devices. So, let us discuss the surface contact devices, the devices which in contact with the skin. That example of such type of devices are surgical tapes, bandages, props that is used in the certain devices. Then external orthopedic braces. So, such type of devices which can be contact with the skin is considered as a skin contact devices. Mucous membrane devices, the devices that contact intact mucous membrane, that is endoscopic, endotracheal tubes, oral swabs or the intrauterine devices. Compromised surfaces or breached surfaces, the devices that contact breaches or otherwise compromised body surfaces, that dressings or the bandages for wound or ulcer or the burns. The devices which is in contact with such conditions that can be classified or considered as a breached or compromised surface contact devices. External communicating devices, this also further divided into the nature of the contact. If the devices that contact the blood path at one point and serve as the conduits for entry into the vascular system, like intravascular sets, extension tubes, transfer sets, etc. The devices that contact the tissue, bone or dentine systems, that is considered as a tissue, bone, dentine devices, or the circulating blood devices, the devices that contact the circulating bloods. Example of these devices are oxygenator, intravascular catheters, dialyzers. Implantable devices, the devices contacting the bone or the tissue and tissue fluids. Examples are the pacemakers, breast implants, orthopedic implants. The blood implantable devices, the devices principally contacting the bloods. Example of such type of devices are the heart valves is one of the example, vascular grafts, the pacemaker electrodes. So, these are the various types of the medical devices based on the types of or the category of the contact of the medical devices with the human body, that can be categorized into three main part, and the duration of the contact, that we have discussed that three types of the duration is prescribed in the standards. The limited exposure, that is the devices is in contact with the human body up to the 24 hours. Prolonged exposure, that is defined as the devices which in contact with the human body between 24 hours to 30 days, and the permanent contact devices that can be categorized if the device is in contact with the human body which exceeded 30 days. The contact includes the single or multiple uses. The cumulative effect of the devices can also be considered as a based on the use of the devices, that also be considered as a permanent contact devices if it can be used multiple times beyond the 30 days. This is the appendix A, that is the matrix. Based on the device categorization, we have discussed that device categorization is based on the category of the devices with the contact of the human body and the duration of the contact. This table gives which mandatory tests are required for the medical devices where there is any biological evolution data or the toxicity data are available. The device is novel in nature or the biomaterial is used first time. In such cases, based on their category, the duration of the contact and the nature of the contact of the devices, these certain test that is prescribed in the standards, that is the cytotoxicity, sensitization test, irritation test, systematic toxicity test, subchronic toxicity study, genotoxicity, implantation test, hemocompatibility test. These are the minimum tests that is required to be carried out by the manufacturers. If you see these standards ISO 10993, if you compare with the US guidelines, in USA certain additional test is also required to be carried out, and this table shows which are the additional test is required to be carried out on the particular devices based on the categorization of the devices. Certain additional test is performed, but this test is as per the requirement of US FDA, and however the manufacturer can do the test based on the available standards, and they can justify after carrying out the risk based assessment of the particular medical devices. The endpoint identified by X or the 0 in annexure A may not be relevant for all the devices in particular category. We have discussed that it is only applicable for the devices where there is no data is available with respect to the material chemical data or the biological evolution data or the toxicity data. In such cases, these test can be, the test prescribed in the annexure A can be carried out, and this is the minimum test. Already I have explained that. The additional test, that as per the US FDA, what are the additional test, that is also mentioned in the another table which is given in the slides. And although this test which is given in the table is not the complete test, it is the minimum test. Beyond those recommended that annexure A, other tests as appropriate that can also be performed by the manufacturers, and also that can be recommended by the regulators to perform that particular additional test which is not mentioned in the annexure A of this table. Now, the basic evolution test we have discussed, that is the cytotoxicity test, sensitization test, irritation or intercutaneous reactivity test, systematic toxicity test, subacute or subchronic toxicity test, genotoxicity test, implantation test and hemocompatibility test. The general purpose for conducting this test, we can just have look the cytotoxicity test, that is performed to determine the cell death or the cell growth inhibition and other adverse event on the cells caused by the medical devices or materials and or their extract, and the details of the test to be performed by the manufacturers is given in the ISO 10993 series 5. Sensitization test, this test is to be used to determine the potential for contact sensitization by the test material or the devices. The standards ISO 10993 series 10, which gives the details of the test which is to be performed. Series 10 also includes the test for irritation test. This irritation test is used to determine the irritant effect of the medical devices or material or its extract or the tissue such as the skin, eye or mucous membrane. The series 11 of ISO 10993 that describes the detail of the systematic toxicity study. Under this study, the acute toxicity study, subacute toxicity study, subchronic and the chronic study that how to perform, how long that test can be performed, that is given. For acute toxicity, that is the to determine the harmful effect of the devices either by single or multiple exposure during the period of the sample less than 24 hours. If the duration is between 24 to 28 days, that subacute toxicity is required to be carried out. If it is 28 days to 90 days, then subchronic, and beyond 90 days, that is chronic study is required to be carried out, and the duration of the chronic studies shall be carried out up to 6 months to 12 months. And series 11 describes the details. Genotoxicity test, ISO 10993 series 3, that gives the details about the genotoxicity test. These tests are used to determine the gene mutations, changes in the chromosome structures, and the number and other DNA and gene toxicity caused by the medical devices or materials and or their extract. Implantation test, this test is used to determine the local effect of an implantable devices or the material or the tissue by implantation procedure that introduce the material or devices into the tissue of the animals. And series 109936 that gives the details of the implantation test. Hemocompatibility test, that is the test is required to use to determine the compatibility of the devices or the component with the circulating blood. And the series 10993 series 4 that gives the details of the hemocompatibility test study. Chemical assessment of the devices, we have already discussed that. Additional chemistry information also required to be carried out by the manufacturer to support the long history of the safe and use the rationales. This chemical assessment is also needed, the devices with unexpected biocompatibility test finding. The device manufactured from the material that intentionally change over a time. In such cases, this chemical assessment is also required to be carried out prior to initiate the biological evolution study. Some devices including the chemical with the known toxicity where the new biologics testing is rarely conducted. In such cases, this chemical assessment is mandatory. New chemical entity used first time or modified material that formulation in such cases, or the novel materials if it is used, then chemical assessment is mandatory prior to conduct the biological evolution test of the medical devices or material. So friends, this is all about the biological evolution. I hope you have now got the general idea about the biological evolution test or biocompatibility test. What are the standards applicable for this? What is the guidelines? What is the risk based approach for selection of the biological evolution test? Categorization of the medical devices for the biological evolution, how to categorize, and the standards, the main standards which is applicable for conducting the biological evolution. So, this is all about these lectures, and before concluding or closing these lectures, let us have some question answer session. Now, the first question, can you explain what is the applicable standards for conducting the biological evolution of the medical devices as per Medical Device Rule 2017? Yeah, in the correct answer that we have long discussion on the standards, that is ISO 10993. Now, another that question ki how many that series of the ISO 10993 is there? Approximate we have discussed. Yeah, tell me. talks that more than 20 standards, 20 series of the standards is available, which covers the biological evolution. Now, the last question, whether the biological evolution test that is not under the purview of this ISO standards, which type of the devices that cannot be, in which the biological evolution test is not applicable. Yeah, the devices which is not in contact to the human body, in such type of the devices this biological evolution test is not required, that is ISO 10993 is not applicable for such type of devices. So, this is all about this biological evolution test. Thank you very much, and another topic we will discuss in subsequent lectures. Thank you very much.